The, the floor is yours, please.
Okay, thank you very much. Okay, so let me just share this. So thank you everyone for coming, and I will just let me change it to a laser pointer. Okay, so hopefully now
Mhm.
you can see my slide and the laser pointer.
Yes,
So
yes, we can
hi
see.
everyone. Okay, great. So hi everyone, I am Karerra. I'm a lecturer in Durham University, and my expertise is in the area of biochemistry and microbiology. So this lecture that I'm going to do today is actually a lecture that I deliver to our level 2 undergraduate students in biology. And this is in a course called microbiology. And this specific lecture is an introduction to the roles of metal ions in microbiology. So I will just basically tell you a little bit about metals and why they are important for life, but especially microbial life. So the objective of this lecture is basically as simple as that on the slide, which is to understand the importance of metals in biology, but we will be focusing on microbiological life. Okay, so when we think about biology, we've probably heard about the different types of ohms. So we know about the genome, which is the collection of all of the genes that are present in the cell. We know about the transcriptome, so the genes are translated into transcripts or RNA, and the collection of all of the messenger RNAs are called transcriptome. And those transcripts are then translated to make proteins. So the collection of proteins in the cell are called proteome. And of course, different proteins can do different activities. They can generate metabolites, they can make lipids, and they can also make carbohydrates. So we also know about metabolome, lipidome, and glycome. So these are all the very, I guess, diverse and important ohms of biology. But what is often, I guess, forgotten is that at the heart of all of these ohms is actually what we call the metalome. So this is the collection of all the metal ions inside a cell. Oops, let me just go back. And if I can, if I suppose if you can remember only one message from this lecture, I want it to be this. I want everybody to know that almost every cellular process or every metabolic pathway inside a cell requires at least one metal binding protein or one metal binding enzyme, and we call it metalloprotein or metalloenzyme. So please remember this. So no matter what you encounter in biology, it is very possible that that particular process will depend on a metal binding protein or a metal binding enzyme. So when I talk about metals, what do I mean? So metals in biology comprise these metals that are quite abundant. So you have sodium, magnesium, potassium, calcium. But biology also can use these metals here that we call the trace metal elements. So these are manganese, iron, cobalt, nickel, copper, and zinc. Some organisms can also use vanadium, molybdenum, and tungsten. So these, whenever I say metals, or whenever you think about metals in biology, these are the metals that we're talking about. These are the metals that are required by all living organisms. Now, this requirement is actually quite high because if you catalog all of the enzymes that are present inside a cell, about half of them are actually metal-dependent enzymes. So this here is a summary of the metal requirement of all enzymes. About 16% is magnesium dependent enzyme. About 8% of all enzymes are iron dependent. So really, there is a huge requirement for metals for the, I guess, for cells, because nearly half of all of the enzymes inside the cells will need a metal ion to become activated. So because nearly half of all enzymes in a cell need a metal ion, that means that if there is too little of a metal in the cellular environment, that means that it will inhibit cellular growth and metabolism because the cells will starve. These metals are essential nutrients, so if there's too little of a metal ion, then the cell will starve, and this will affect their growth and development. This is an example of an important cellular process or metabolic pathway that requires a metalloenzyme to occur. So you may be familiar with this cycle. This is the TCA cycle, which is a major, I guess, pathway for carbohydrate metabolism in many living organisms. And these are the different enzymes in blue. These are the enzymes that catalyze the TCA cycle. And I have highlighted in red boxes here. These enzymes that I have highlighted are all metalloenzymes. These enzymes require iron, specifically in the form of iron superclusters, which I will talk about a bit later. But these enzymes require iron in the active site, and therefore these enzymes cannot function if the cells do not have enough iron. And if the cells do not have enough iron and these enzymes don't work, that means that this TCA cycle pathway will stop working, and that means the organism cannot metabolize carbohydrate anymore. So this is one example of why, of how a metal ion is important for a cellular process. Now, there are also other types of processes that require a metal ion. For example, if you think about respiration and energy production, that process requires multiple metalloenzymes, and these are metalloenzymes that need iron, calcium, and also copper. If you think about the ribosome and the translation of mRNA to proteins, that process requires a lot of zinc and also sometimes manganese. If you think about transcription, so the generation of mRNA from DNA, that also requires quite a lot of zinc. So anything that happens inside a cell, most likely it will be metal dependent. Now, there are different types of metals, metal cofactors. So metal cofactors are basically the metals that bind to proteins and enzymes. These metals can be present in the ionic form, so it can be just as simple as iron 2+ ion or sodium plus ions, or the metal ions can be in some variety of structures. So you may be able to recognize here the structure of heme. So this is, of course, the molecule that is responsible for the red color of your blood. So this is heme is contained in the protein hemoglobin, and this is the major oxygen carrier in your blood. So this is an example of how a metal ion can be embedded into an enzyme, but in the form of a cofactor rather than in the an organic cofactor instead of a free metal ion. There are also structures called the iron superclusters. So you can see that it is a cluster of sulfurs and iron. And these are typically found in the TCA cycle enzymes, for example, that I showed in the previous slide. There's also this structure called the cobalamin. So this is actually the precursor to vitamin B12. In this case, the metal center is actually a cobalt metal center. but the organic structure here is called a serraheme or a porphyrin. This is almost like the structure that we see here. So this is a porphyrin ring, but it's just that the decorations around this ring is slightly different. And also the metal center is cobalt instead of iron. And finally, we also have this structure called the molybdotierin. So this is a cofactor that contains molybdenum as the metal center. Right. So yes, so metals can be bound to proteins and enzymes in these types of different forms. Oh, right. So how do proteins then how do these metal ions then attach to proteins or enzymes? Well, they usually attach to the side chains of these amino acids, so methionine, cysteine, histidine, glutamate, and aspartate. These are usually the amino acids in proteins that can bind to metal ions. So, for example, this here is a crystal structure of a zinc finger protein. So this zinc finger protein is important for nucleic acid metabolism, so DNA and RNA. And you can see in the structure here is quite a small protein. You can see two beta sheets. You can see the alpha helix here, and you can see the zinc center here in green. Now, the zinc center binds to the zinc finger protein because it coordinates with the sulfur side chain of the cysteine. There's one cysteine there, another cysteine. And the zinc also coordinates with the imidazole side chain of one histidine here and a second histidine there. So this is how zinc can bind to a zinc finger protein. Another example is this. So this is hemoglobin. Of course, this is the major oxygen, well, the only oxygen carrier in your blood. This here, you can see the alpha helix of the hemoglobin here in green, and you have the heme cofactor in this structure. You have the iron center shown here, and the iron center is coordinated by this imidazole ring of the histidine from the hemoglobin. So this is another example of how a metal ion or a metal cofactor can bind to a protein. Right. So what are the different roles of metal ions in biology? How come half of all enzymes need metal ions? Well, what do the enzymes do? And what do the metals specifically, how do the metals specifically help these enzymes achieve their function? So there are four main roles of metal ions in biology. First, they can act as second messengers in signal transduction. They can also act as a charge carrier that can help set up an electrochemical gradient across the cell membrane. Metals can help in the formation of the final protein or enzyme structure, so it may help with protein or enzyme folding. And finally, metal ions may also play a catalytic role. It may provide a protein with a particular reaction or catalytic ability. So I'm going to go through these examples one by one. The first example is this, which is a metal ion calcium acting as second messengers in signal transduction pathways. So this here simply summarizes how calcium can participate in various signaling cascade or phosphorylation cascade in the eukaryotic cell. So the differences or changes in calcium concentrations inside the cell can actually trigger different phosphorylation reactions, and this will then lead to a particular cellular outcome. So this is basically the role of an example of the role of a metal ion as a messenger in signal transduction pathway. The second example is a metal ion acting as to set up an electrochemical gradient across the cell membrane, and this electrochemical gradient can be used to drive work. So this role of a metal ion in setting up an electrochemical gradient is actually very similar to the role of protons in setting up an electrochemical gradient or a proton motive force. But instead of protons, this electrochemical gradient uses a metal ion, and this metal ion is typically sodium, potassium, or also calcium. One example here is the structure of a bacterial flagellum. So blood flagellum is the, I guess, the structure in a bacterial cell. It sits on the surface and it helps the bacterium actually rotate and move around. So here is the schematic. So this is the flagellum. So it's like a tail, and this flagellum basically rotates, and this rotation helps the bacterial cells swim around in that environment. So here is the structure of the entire flagellum motor. So the base of the motor is the part that rotates. It kind of turns around. You can see it in the scheme here. So the motor actually turns around, and this rotation of the motor, and therefore the rotation of the flagellum, relies on a sodium electrochemical gradient across the bacterial membrane. So this is an example of a metal ion providing the energy to drive certain types of work in a cell. Another example for the role of a metal ion in biology is that it may help a protein or an enzyme to adopt the correct final structure. By doing that, the metal ion can promote protein folding, or it can stabilize the protein structures. So an example here is the role of zinc in zinc finger proteins. So as I said earlier, zinc finger proteins are important for nucleic acid metabolism. So it's important for translation and transcription. And it aids DNA and RNA polymerases in their function. So here is a structure of a zinc finger protein in blue that wraps around nucleic acid. So you can see the DNA in the middle, the double helix in the middle. So that's the DNA, and we've got the blue structure here wrapping around the DNA, and that is the structure of the zinc finger protein. And you can see the zinc in the green circles here. So the zinc finger protein only adopts this structure because zinc is bound to the protein. If the protein does not have zinc, then it will not have the correct structure. And if it does not have the correct structure, then it will not be able to bind to nucleic acid. And this is just another example. In this case, it's a zinc finger protein wrapping around an RNA instead of a DNA. And last but not least, there is also metal ion also play a role in as a catalytic center. So there are two types of the roles of metals in a catalytic center. The metals can act as a non-redox active catalytic center or enzyme cofactor. So in this case, the metal and the enzyme do not catalyze a redox reaction, but they still do catalyze a reaction. And one example of such an enzyme is the lactamases, especially the metallolactamases. And these enzymes, you may know, these are responsible for antibiotic resistance against beta-lactams or penicillin. So here is the structure of the protein NDM1. So this is an example of a metallolactamase that is currently a huge problem, specifically in India. This is called New Delhi metallolactamase enzyme. You can see the structure here in blue, and the active site pocket is here. This here is a zoom in of the active site pocket. We can see the beta-lactam molecule. So this is a penicillin molecule binding to the enzyme. This here, the kind of four-membered ring here, this is the beta-lactam ring. You can also see the zinc centers in pink. There are 2 zinc centers in the enzyme. So in this case of metallolactamases, the zinc centers actually help to bind to the enzyme substrate, which is the beta-lactam or penicillin. And by binding to the penicillin or the substrate, basically zinc here is acting as a Lewis acid. And this essentially helps allow the enzyme to break this beta-lactam ring. This beta-lactam ring breaking is not a redox reaction. It's simply a hydrolysis or addition of a water molecule. And zinc atoms here are acting as a Lewis acid that helps the hydrolysis of this beta-lactam ring. But this is not a redox active reaction. But an example of, the more, I guess, common role of metals as a catalytic center is the role of metals as electron carriers or redox catalysts. So they catalyze redox reactions. And this is metals are particularly good as redox catalysts because some metal ions can exist in multiple oxidation states. So for example, here copper can exist inside cells in biology in the copper 1+ oxidation state and a copper 2+ oxidation state. And these 2 species can basically interconvert between one another. So copper 2+ can be reduced to copper 1+, and copper 1+ can be oxidized to copper 2+. And this redox reaction between the 2 copper species can be coupled with the oxidation of an electron donor or the reduction of an electron acceptor. And if both of these molecules happen to be something inside a cell, then it essentially means that copper can catalyze something, I guess, useful inside a cell. And one example that I want to highlight here is the role of metals in 2 enzymes called the superoxide dismutase and catalase. So both of these enzymes are redox catalysts. They catalyze redox reactions, and specifically, they detoxify reactive oxygen species. And the reaction shown here goes like this. So we start with superoxide. This is actually both oxidize and reduce at the same time by superoxide dismutase to make oxygen and hydrogen peroxide. And hydrogen peroxide is then further reduced to make oxygen and water by this enzyme called catalase. Both of these enzymes, because they can detoxify a reactive oxygen species, they are often very important for bacteria or microbes. And this is because for these bacteria, they can often encounter superoxide or hydrogen peroxide when they come into contact with for example, an innate immune cell from humans or animals. So if a bacterial species happens to be a pathogen and this pathogen infects a human cell, the human cell can release superoxide and hydrogen peroxide, and the bacteria can then defend themselves by using these two enzymes, superoxide dismutase and catalase. And both of them are metal-dependent enzymes. So here is just a bit more detail for the specific reaction mechanism for how superoxide dismutase works. So in the resting state of the enzyme, normally copper is found in the oxidized form. That oxidized form of copper will react with the first molecule of superoxide to make oxygen, and in the process, this copper center becomes reduced. This reduced copper center then reacts with a second superoxide molecule, and this superoxide molecule is reduced to make hydrogen peroxide, and in the process, this copper center is reoxidized back to copper 2+, and the cycle keeps going until all of the superoxide has been depleted or used up. So here are some structures of superoxide dismutases that have been, I guess, identified in biology. So these come from different organisms, some bacterial, some plants, some fungi. And there are quite a variety of different structures. You can see in this case, this superoxide dismutase binds copper and zinc, and it seems to be a homodimer. This superoxide dismutase binds nickel, but this is actually a hexamer. You can see 1, 2, 3, 4, 5, 6 proteins in there. And then we also have manganese superoxide dismutase and iron superoxide dismutase. So even though all of them have slightly different structures and bind different metals, they all carry out the same reaction, which is the detoxification of superoxide to make hydrogen peroxide. I should also note that the use of metals in a particular enzyme is specific. So for example, this superoxide dismutase only works with copper and zinc. It does not work with nickel. In order for an superoxide dismutase to work with nickel, it has to have this structure. And this enzyme works only with nickel, but not with copper and zinc. But both of these enzymes can catalyze the same reaction. Now, only metals can do this job, can react with superoxide, because the redox potentials of the metal centers match the redox potentials of the reaction of superoxide reduction and superoxide oxidation. The importance of metals in this reaction is perhaps, I guess, highlighted by the fact that we have not ever discovered a superoxide dismutase that does not have a metal. All superoxide dismutases that have been found in biology, all of them have a metal center, because metal centers are fantastic redox catalyst for this reaction. This is the other enzyme, so catalases, which detoxify hydrogen peroxide. Again, there are a variety of structures. This one on the left here is an iron-dependent one, and actually the iron here is found in the heme form, whereas this catalase requires manganese. So like I said before, because the use of metals is specific, so this particular catalase can only work with manganese. It does not work with copper, it does not work with zinc, or it does not work with iron. And likewise, this catalase here only work with iron heme, and it does not work with the cobalt cobalamin structure that I showed earlier. And just like the superoxide dismutases, only metals can catalyze this reduction of hydrogen peroxide, and we have not ever seen or discovered a catalase in any living organism that does not have a metal center. So there are no metal-free catalases has ever been identified, ever. Okay, so I have just gone through the four key functions of metal ions in biology. So first, metals can act as a signaling messenger. It can also help set up an electrochemical gradient across membranes that can be used to drive work. It can help proteins fold to the correct structures. And finally, it can help enzymes perform catalysis. So now this means that metals are important nutrients for all life. But now I am going to focus specifically on bacteria. Bacteria, just like all other cells, they all need metals, but they must take up these metal ions against a concentration gradient from the extracellular external environment. Metals, as you know, cannot be synthesized, so all living cells, including bacteria, must take up these metal ions from the environment. Now, in this graph here, this comes from a paper. You can look it up if you're interested. But basically, this measures how much metal is present in two types of samples. So the samples are cells. In this case, it's E. coli cells. And the other sample is the culture medium. So in this experiment, the scientists have grown E. coli in a medium. And what they've done then is to separate the cells from the medium, and then they measure how much metals are present inside the cells and how much metals are found in the culture medium. And what they found is that for the culture medium, these are the graphs with the vertical lines. For the culture medium, the levels of metal ions, almost every single metal ions, all of them are less or lower than the amounts of cells that they measure, than the amount of metals that they measure inside cells. So basically, the scientists found that there are a lot more metals inside a cell than there are metals in the environment. So this actually means that the environment doesn't have a lot of metal ions, and the cells accumulate these metal ions inside those cells. So this can only mean one thing. That means that cells must move against a concentration gradient. So, metals must move against a concentration gradient. Cells must take up metals from the environment, but the environment has a lot less metals than the cells. So that means metals must move from an environment of low metal concentration to an environment of high metal concentration. And the only way that this can happen is if cells use active uptake. So active uptake is energy-driven uptake processes. So how does active uptake of metals occur? Here I have the schematic of a diagram of a gram-negative bacterium. So gram-negative bacterium, of course, has two membranes. The first one is the periplasmic membrane that faces the outside of the cell. And the second one is the inner cytoplasmic membrane. And then we have the inside of the cell down here. In order to cross these two membranes, metals cannot simply diffuse through the membranes because the lipid bilayer of the periplasmic membrane and the cytoplasmic membrane, that lipid bilayer is extremely hydrophobic and also very tightly packed together. So metal ions cannot cross the outer membrane and inner membrane simply by diffusion. The only way that cells can take up metal ions if via transporters that allow the movement of metals across these membranes. The other reason why metals do not just move by diffusion is because, as I said earlier, outside the cell, there is actually a lower concentration of metals compared to inside the cells. So that means if metals have to diffuse, it will be diffusing against a concentration gradient, which does not happen. So metals can only be taken up by cells if there are transporters that allow those metals to cross the lipid bilayer. So there are outer membrane receptors for metals, there are inner membrane receptors or transporters for metals, and the key requirement for both of these transporters is that they must be driven by energy. And by energy, I mean these transporters require the hydrolysis of ATP, or it requires the proton motive force of the proton gradient across the membrane. Right, so those are the two different types of transporters. A primary transporters uses ATP hydrolysis, whereas a secondary transporter uses proton motive force. And I just would like to highlight that although we are currently talking about importers or uptake, there are actually also transporters that catalyze metal movement in the opposite direction. So there are also metal exporters, but we're not going to talk about that today. Okay, so now I am going to go through one major example of a very widely distributed class of a metal transporter. And this class is called the ATP binding cassette or ABC transporter. And the name kind of already gives you a clue. These transporters are driven by ATP hydrolysis. So this is just a schematic that explains how that transport occurs. So there are three components to this ABC transporter. There is the extracellular or the soluble substrate binding protein. So this is the protein component that will bind to the substrate. In this case, this substrate is a metal ion. And once the substrate binding protein binds to the metal, it will then bring that metal to the transporter. And the transporter is composed of two parts. The first part is the transmembrane domain. So this is the part of the protein that crosses the membrane. And there is another part of the protein that is found inside the cytoplasm, and this part here is the ATP hydrolysis domain. So the substrate binding protein binds to a metal ion, then delivers this metal ion to the transmembrane domain. Once that metal is bound to the transmembrane domain, this actually triggers a change in the conformation or in the shape of the protein. And this then promotes the first ATP hydrolysis. So ATP hydrolysis makes ADP, and this triggers another change in the structure of the protein or a conformational change in the shape of the protein. And that triggers the second ATP hydrolysis step to make ADP. The second hydrolysis step triggers another change in the shape of the protein. In this case, the protein or the transporter, the change in the shape is quite dramatic. So from being initially open to the outside of the cell, so this is a V shape, now it triggers a change so that it is now closed to the outside of the cell, but open to the inside of the cell. So this then allows the metal to be released into the cell, and the release of metal into the cell, release of the substrate binding protein, and the whole system resets back to the beginning. So if you were paying attention, then you would probably hear me say a change in the structure or a change in the shape and the conformation of the enzyme. And this is the key feature of transporters. The energy, in this case ATP, drives this change in conformation or change in structure, and that change in structure is what drives the import or the internalization of metal from the outside of the cell to the inside of the cell. So that was what I showed earlier, was just a schematic, but of course proteins have structures. So these are just two examples of ABC cassette importers. You can see that the structures are perhaps slightly different between each other, but you can hopefully recognize the, I guess, general features. So you can see in the red here on the left and the red on the right, both of these will be the soluble substrate binding protein. And then you can also see the transmembrane domain here and the transmembrane domain there. Maybe this transmembrane domain is a lot bigger compared to this one, but they're both kind of the same types of transmembrane domains. And finally, you can also see the ATP hydrolysis domains here inside the cytoplasm, and likewise it's here. So even though the shapes might be slightly different, and even though they might transport different types of molecules, but hopefully you'll be able to recognize the general features, and they all work via this mechanism. And just like before, when I said that the metal ions are specific for the enzyme in the case of superoxide dismutases, this is also true in the case of metal transporters. So metal transporters that take up, for example, manganese will probably not take up heme iron or copper, for example. So one transporter is specific for one metal ion. So this is an example of a major class of the primary active transporter, which is ATP driven. Now, well, yes, there you go. So this is the keyword. This conformational change or change in the structure of the transporter is what is important in driving the direction of metal transport. So here is an example of a secondary active transporter. In this case, is driven by the proton motive force, so the chemiosmotic gradient of protons across the membrane. So this is another major family of transporters, and these are called TONB-dependent receptors or transporters. There are a few components of TONB-dependent receptors. So the main components are shown on this slide. So there is a beta barrel component, and this beta barrel is embedded into usually the outer membrane of bacteria. And you can see a hole here inside the beta barrel. And inside the hole, there is what we call a plug domain. So the structure of that domain is shown here. And in this receptor, that plug domain sits inside the beta barrel. Now, this plug domain will bind to the metal ion or metal cofactor that it is supposed to transport. So in this example, the substrate here is actually an iron complex called ferrichrome. So it's transporting ferrichrome. So you have the ferrichrome binding to the plug domain. And as soon as that ferrichrome binds, which is shown here, then what happens is that it will trigger a change in the shape of the plug domain such that there is now this tail that is released, and this tail is kind of released and it exits outside the beta barrel. So you can see it better here perhaps. So this is before it binds to ferrichrome. After it binds to ferrichrome, you can see that the structure here is quite different, and importantly, it exposes this kind of tail. And this tail here we call the Tonby box. Here is a bit more detail about what that Tonby box does. So in this case, the transporter is transporting vitamin B12. So you can see the plug domain here. Vitamin B12 binds to the plug domain, which sits inside the beta barrel. And as soon as that molecule binds to the plug domain, Tonby box is released. And this Tonby box is released into the other side of the membrane. And this box will meet Tonby. These two things will meet. Now, Tonby is a protein that is embedded, actually, attached to the inner membrane of the gram-negative bacteria. And this Tonby molecule is part of a, I guess, multi-subunit structure. And this multi-subunit structure will actually allow Tonby to bind to the Tonby box. You can see the binding here. And once this binding occurs, there's a signal that gets transferred down to the inner membrane. This is again a conformational or a structural change. And then this inner membrane modules or subunits will use PMF, so the proton motive force, to mechanically pull or yank this Tonby down. So basically, this downward movement will essentially move the plug out of the beta barrel, and this allows this molecule to then enter into the periplasm. So this is another example of how metal uptake can occur, and this is driven by the proton motive force. So really, that is all I have for today's lecture, and I am hoping that by now you would, I guess, appreciate that metal ions are very important in biology, and I hope that you will understand what are the different functions of metal ions in biology. Of course, there are 4 of them. So number 1, as a signaling messenger or signaling molecule. Number 2, as to set up electrochemical gradient. Metals can also help proteins fold to the correct structure. And finally, metals can act as catalytic cofactors in enzymes. And why they are important for bacterial physiology? Well, because all of the cellular processes not only in bacteria, but also in all living organisms, a lot of the processes, for example, we have talked about the TCA cycle in the carbohydrate metabolism pathway. Those processes require a metal-dependent enzyme. And finally, I lost it from this slide, but finally, I hope that you will also appreciate how metals can be taken up into a cell, and this will be via active transport, so energy-driven transport. Metals cannot simply diffuse into cells for reasons that we have already discussed. Instead, metals must be taken up by specific transporters, and these transporters can be driven by ATP or by PMF. And the key point is that at every step, there must be a conformational change that allow for the metals to move from one side of the membrane to the other. And actually, before I end, maybe what I want to do, let me just stop sharing for a moment. What I want to do is actually to show you a video of how the TONB receptor works, because it is actually very cool. Let me see if I can find it on YouTube. Because this, I think it's easier to see a video than me trying to describe it with my pointer. Oops, accept. Oh no. Oh, right. Okay, let me see if I can find it. Okay, here we go. Hold on. How do I need to share my screen again. Share screen, share screen, share. Hopefully you will. Ah! Sorry, it's advertisement.
Oh, no, it's okay.
Okay, here we go. There's no sound, so that's fine. So this is just an example of why metal ion uptake can be quite important, especially for pathogens, because the pathogens must take up iron, in this case from the blood, and they use a TONB-dependent transporter to take up iron. So here we go. We can see. So transparent is a molecule from the blood. So this here is a TONB-dependent transporter. This is a TONB box. That's TONB coming in, and PMF basically drives. Yep. Yeah, so that's an example. So if you look up this video, hopefully you'll be able to to see it in more in more detail. Okay, so that is everything I have. Hopefully that has been understandable for everyone and useful.
Yes, Karerra, thank you very much. It's very interesting, especially I think for my colleagues, for my students, and it's mm, as we all understand that the similar mechanism not in only in the microbial cells, and it's the same biochemistry pathways, it's in our own bodies too. So
Yeah.
I think we have the same problems. But Carrie, I want to ask you some interesting question. How do you think why exactly these ions are used by the biological life on the earth? Why but not another maybe
Yeah.
elements. It's very interesting because I think from my point of view, maybe it's just like an evolution on the Earth and the number of these ions in the environmental. I think
Yes. Yeah.
it's the simplest answer, but maybe some another mechanisms, and also it's. Mm, Clearly seen that the main of these ions is they have two electrons, and
Hmm.
I think maybe sometimes they can uh, be like different cofactors. So maybe in the one enzyme can one enzyme use the not only
Yeah.
one ion or maybe two or three with the appropriate.
Yeah. No, yeah, you're absolutely right. Why these metal ions and not others? Yeah, most of it is because of whatever metals are available in the environment and in the course of billions of years of Earth. I guess
Mhm.
these are the metals that are available. So yeah, absolutely right. You might be interested to know that there are also I'm just gonna share my screen again. There are also actually recently people found that some bacteria from like deep ocean
Mhm.
can actually use the lanthanide. So there are bacteria that can use these metal ions for their metabolism.
Okay, I think
So
because maybe deeply on the bottom of the ocean, it's not so available another ions. Uh,
exactly, yeah,
but you see that I think it must be like a sign that the evolution never stop, and maybe in the future we can some
yeah,
effects of some reactions, and using maybe if we changes these ions, maybe the it can improve, maybe
yeah,
do faster some
yeah.
reactions, and how do you think this works can be done by humans to try to change these bonds inside enzymes and between enzymes and ions.
Yeah, no, there are a lot of people trying to do that. So there are a lot of people trying to make enzymes work with, new metal ions. So with mercury or with lead or with cadmium, things
Mm-hmm,
that we usually don't think about as useful. We actually think about them as toxic, right? But there are definitely a lot of work out there trying to make them useful somehow. I don't know much about what has been happening, but there's definitely people trying to do that. But I think um there are different approaches, right? You can either try and design these and see, oh, maybe if I make it like this, it will have this activity, or you can just let the protein or bacteria evolve
mm-hmm.
themselves, I suppose. So yeah, so yeah, absolutely. There are definitely lots of work out there trying to do that.
Окей, thanks. А шановні учасники, можливо, в когось є запитання? Зараз є шанс їх задати.
Можу я задати?
Да,
Uh,
Крістіна, ви зможете
mhm.
англійською, да?
Of
Мгм.
course, yes. Can I add some information and
Yeah.
then ask a question? Thank you for your informative lecture. I am not a biochemist. I am an ecologist and
Okay.
ecologist. I am ecologist, and it was very interesting to listen to your lecture because 5 years ago, when I was studying environmental microbiology, we studied the effect of certain concentrations of non-essential heavy metals such as cadmium and no, also copper and zinc on inhibiting carotenoid synthesis.
Oh.
we studied Rhodotorula genus, and after that we recommended using some sensitive strains for bioindication of heavy metals in wastewater. It is a very interesting work, and I am currently studying medical microbiology because I work in Zaporizhia State Medical University, and I was interested to hear about the movement of bacteria, and I think your slide showed a flagellum of a gram-negative bacterium. Yes, maybe it was E. coli.
Yeah.
But I'm wondering how spirochetes move, because they have an endoflagellum inside the periplasmic space.
Yeah.
Does a proton pump also participate in their movement?
So yes, so because the flagellum, even though yes, it is in the periplasmic space, I'll show you the structure again.
Mhm.
The whole thing is actually anchored or embedded in the inner membrane. So if you can
Mhm.
see this one, so
Yes, yes, we see.
the flagellum itself is on the periplasmic membrane, but it's connected. Oh, actually, this one here is better on the right. Oops.
I understood.
So
Okay.
yeah, the flagellum is on the outer membrane or periplasmic membrane, but it is connected to the motor in the inner membrane. And because the proton motive force is in the inner membrane, right? And also the sodium force is also in the inner membrane. But because this motor is in the inner membrane, it can still move the flagellum.
Okay, sense. But I have an interesting questions
Yeah,
about in English maybe passive absorption, because a long time ago I read an article that some yeasts such as Rhodotorula aurantica, Rhodotorula
yeah.
rubra can passively absorb many heavy metals from water. Even dead cells can do this. And
Oh,
how do you explain the absorption of metals on dead cells? And we use this yeast for maybe water treatment.
is it do you know if the cells are only absorbed onto the surface or do they go into the cell?
Maybe the surface, on the surface of the membrane, chitin, I don't know. And no, I read this article that about some yeast can absorb heavy metals on the
Yeah,
surface of
yeah,
their
yeah,
cell wall. Mhm.
yeah. Surface absorption is
Adsorption,
definitely,
yes.
yeah, definitely common. But if they're dead, then I think people have actually confirmed dead cells don't take up, so that the metals don't get all the way into the cytoplasm, for example.
Mhm.
But there are, it is also possible that if, for example, the membrane, oh no, if the membranes are leaky, but then if the membranes are leaky, then they will leak metals out, not metals in. Um,
Mhm.
Probably, yeah, I would say that probably the absorption will be on the surface and
Mhm.
not inside the cell.
Mhm. Okay,
Yeah.
okay,
So
thanks.
that would just be simple electrostatic or charge interactions, right?
Right.
Because the metal ions are usually positively charged and fungal or bacterial membranes are usually negatively charged,
Mhm.
or surfaces are usually negatively charged.
And peptidoglycan also maybe, yes.
Exactly. Yeah. Yeah.
Mhm.
Yeah.
Mhm. Okay, thanks.
Thank you.
Окей, another questions maybe? Є ще якісь запитання, можливо, в наших учасників? Ну, якщо запитань нема, тоді ми будемо дякувати нашій лекторі. So, Karerra, thank you very much. You know, it's
Thank you.
your lecture, it was very informative, very good, and you know, I think maybe it's just like a start point for of our some of our participants to deeply more in this topic, in these themes, because you know, actually, especially, you know, for our for our region, for our city, we have a very big, a very large metallurgical industry, and it's a problem with metals, with the heavy metals, but
Contamination, yeah.
yes, and that's why in our region it's very interesting work for us, and we looking as a ways to treatment of our lands, for our waters from the heavy metals in the first step, but this all mechanisms may be the same, and it's very essential to know how we can cope the problems with the uptaking and maybe in some storage, even in biological banks, the metals. And also it is very interesting. So we have an atomic station here near our town. So the elements that have an radioactivity, it's also very actual for our region, and also it is maybe the same mechanisms and for these ions too, and I think it's very important work in the future, and
Yeah, possibly.
yes, and I hope, Career, that it's not your last time in our university,
Yes, I
and
hope so too. Yeah, I hope
and
so
we
too.